Welcome to the MIT Center for Art, Science, and Technology's 2021 Symposium, Unfolding Intelligence, the Art and Science of Contemporary Computation. CAST was established in 2012 with the goal of building and building on connections between the worlds of art, science, and technology. This is the third in a series of symposia that CAST has convened since then, and as with its predecessors, we bring together artists, scientists, engineers, and humanists from within MIT and from the world at large to discuss areas of rapidly evolving research and urgent social relevance, and to find in that dialogue stimulation, confirmation, provocation, intersection, and, we hope, common purpose. At MIT, CAST partners with departments, labs, and centers to integrate the arts across the curriculum, to enrich and encourage artistic collaboration, and to provide support to faculty and members of the MIT community as they pursue their own artistic practice and/or research. In addition to symposia like this, CAST facilitates the sharing of this creative work beyond the Institute by producing concerts, exhibitions, and publications, and making them available to the public. So, thank you for being with us today. We hope you will join us throughout the week at virtual events addressing the aesthetic, technical, and critical issues pertaining to artificial intelligence and computational media. We also look forward to seeing you on Friday, April 9th, as the symposium culminates with a live interactive event to which all attendees are invited and in which you can join presenters and artists in breakout rooms to explore hidden threads between all that has been discussed this week.
I understood. I understood.
Welcome to Unfolding Intelligence, the Art and Science of Contemporary Computation. Thank you for joining us and for bringing your curiosity and attention with you today. Deep time and intelligence, unfolding models, open systems, bias in AI. Even in this sheer titular variety, I hope you might hear whispers of the ambitions afoot. Could the discussion of artificial intelligence be reorganized by the thoughts and work of those who question AI? Who cautiously embrace and reshape it to their needs and from particular perspectives. Those who work with and through it, grounding our ideas about and response to AI in such practices that operate within the orbit of these situated bases of reflection. Perhaps then, rather than being strung along by stories about inevitable outcomes, impacts and profits, singularities, etc., all reasoning that assumes the essence of AI is a sure thing, that its effect on us and our role in shaping it are readily apprehensible relations with a stable thing, a predictable future. Relieving ourselves of that overexposure of AI to intellectual sureties, we may sense a moment in which an instrument of optimization and automation becomes a medium open to reuse and comment, tinkering and informed reflection, even a generator of surprise results. Deep time and intelligence, the portion of the discussion at hand. In this panel, we will see that AI can help us to consider fundamental questions about how sensations, sounds, movements, choreographies of form and matter take shape in time, gain coherence as objects and structures through training and learning, recursion and iteration. Deep time, yes, in terms of the evolutionary timescales at work in growth and form from the diatom to the social, but also deep as in downward to the nano and minute intervals of the fold of a protein or a crack in a crystal lattice. AI seems to assist us in traveling to scales at which primordial logical formalisms operate in dimensions and through structures that defy our concepts and intuitions, but perhaps not those of feature detection capacities exacting computations can process. Holly Herndon, artist, composer, performer, has built and conceptualized for us a way of thinking music in the context of a long durée of ritualized entrainment. Gary Tomlinson, Yale musicologist, provides connections between the story of evolution and the unfolding of AI research that few could account for, given the myriad methods and fields of research that he has been able to synthesize, building connections between humanities and sciences for us. Marcus Buehler, material scientist here at MIT, challenges us to think of AI as something capable of detecting homologies of structure across scales of existence, perhaps even giving us new ways of thinking about universals across scales. Antonio Torralba, computer scientist with CSAIL here at MIT, moves not between scales but across modes of sensation, using AI to link sound to vision and movement to sound, proposing a multi-modal sensory field for AI. I shall withhold any further attempt at synthetic remarks at present and listen now with you all to the thoughts of this wonderful group, inviting you to listen with me for hints of how AI might be providing new insight into form and structure, cohering at divergent length scales, time scales, and between modalities of sensation and varying protocols of entrainment. It is important also now to acknowledge that it is not we, the speakers, nor MIT who claim authority over or seek final say regarding the deep or any ultimate meaning of ancient ancestry. MIT acknowledges indigenous peoples as the traditional stewards of the land and the enduring relationship that exists between them and their traditional territories. The land over which circuits sustaining MIT send their signals to your screens is the traditional unceded territory of the Wampanoag Nation. We offer our partial perspectives in acknowledging that we have much to learn, and that an acknowledgement such as this is only a small first step towards remembering and working through the painful histories of genocide and forced occupation that the Wampanoag Nation suffered. Land on which many diverse Indigenous groups gathered since time immemorial. My name is William Lockett. I'm the current Mellon Postdoctoral Fellow with the Center for Art, Science, and Technology. You'll see me at the Q&A, leading the discussion and also taking your questions through Twitter. Thank you and see you then.
Good morning, afternoon, or evening. Thank you for watching. I'm Gary Tomlinson. Provocative titles are meant not merely to provoke, but to draw us in. And one of the more compelling instances I've come across recently is this one from a 2020 article by J. Mark Bishop. Artificial intelligence is stupid and causal reasoning won't fix it. The essay opposes computationalism, the idea that human intelligence is a strictly computational affair, and it argues that AI, since it is a computational affair, can't duplicate the feats of human-like understanding. Bishop's article is one in a long line opposed to computationalism, both as an approach to the human brain and as a way of making a machine that might act like it. And he takes us on a quick tour of some of his predecessors, from Gödel's incompleteness theorem, as developed by John Lucas and Roger Penrose, to John Searle's Chinese room, riffing on the distinction of formal syntax from semantics. Then, building on Hilary Putnam's idea that all open systems are, in effect, finite state automata, Bishop reasons that equating mind with computation leads inevitably to panpsychism, to the assignment of mind to every open system, to a rock, for example. What worries me about much writing of this sort is its proliferation of key words that are rarely defined as clearly as they need to be. Bishop's review runs through a litany of them. It's true that he mostly avoids the vaguest terms, mind and consciousness. But here are some difficult words he does rely on that call out for unpacking but receive little. Understanding, as in the human-like understanding that automata don't achieve. Or meaning, as in the meaning of words computational machines can't grasp. Other keywords that pop up are synonyms for meaning. Semantics and content both appear in Bishop's discussion of Searle's Chinese room argument. Two more difficult keywords are experience and salience. Is salience dependent on experience, or a part of it, or a generator of it? What is experience, and how does salience come about? this lexical thicket, meaning is for me the key word of keywords, and we can expand the list of closely related terms beyond Searle's content and semantics. Reference, representation, aboutness, intentionality, just to give 4 examples. In my remarks today, I want to make 2 observations about meaning and its synonym cluster. Observations that seem to me relevant to the possibilities of a strong AI or an AGI, artificial general intelligence. The first observation concerns the foundation of meaning, which I locate in semiosis, that is, in sign making. The theory I draw on here is generally humanistic in orientation. The second observation concerns the origins and extent of the meaning that arises from semiosis, and here my theoretical base is in evolutionary science. To look ahead, these observations will lead me toward a couple of general caveats regarding machine intelligence. First, in order to see the challenges of designing a machine capable of understanding meanings, we need a firm grasp of the nature of meaning itself. This requires a distinction that I regard as fundamental and ontological between information transmission, a basic process to all life forms, and sign making, a particular kind of information transmission restricted to a very small proportion of those life forms. Information transmission of the first sort, no matter how powerful or elaborate, can't lead to meaning, content, or aboutness because it can't create signs. Only a machine capable of semiotic information could create or understand meaning. Second, once we see the distinction between information in general and the special case of semiotic information, we discern more clearly the evolved capacities that enable the creation of signs and hence meaning. These are not identifiable traits possessed by particular organisms, not adaptations selected for particular functions. Such selection for is not how natural selection works. It's not an optimization problem, notwithstanding Dan Dennett's decades-long effort to make us think it is. The capacities to create meaning are instead ongoing processes of negotiation with an external world, and so they can't be identified as discrete properties of an organism. Processes, not properties. This is a motto that should hang above the door of every evolutionist. It already hangs above the doors of many. These processes are signaled in the radical niche construction of my title. Okay, so much for looking ahead. Let's start with the distinction of non-semiotic and semiotic information. The first could be called information pure and simple, or Shannon information, but I'll call it causal information, since it's a matter of causal covariance, as Jerry Fodor once put it. Its transmission along some kind of channel brings about some corresponding effect at the receiving end. Information of this sort is a relational thing. The channels can feed back from receiver to sender. They can be networked in parallel and serial fashion, doubled and redoubled again, and hierarchized in any number of routines and subroutines. But the fundamental relationality of covariance remains. All living things, even the simplest bacterium, are complex miracles of this kind of information processing. But their information in the vast majority of organisms is not about anything. It's without content or meaning, a sheer causality that makes things happen from the molecular level on up in scale through organismal and ecosystemic levels. It brings about correlated difference in the complex systems at work at all these levels. Semiotic information is something different that arises on the foundation of causal information. It depends not on systems of relations alone, but on a relation to a relation, a meta-relation. This requires something more complex than the Shannon model of transmitter, channel, and receiver, and something different even from the idea of a hierarchy of routines. It requires something so complex, in fact, that it has emerged only in a tiny percentage of the life forms our biosphere has known. While all life forms are processors of causal information, very few are creators of semiotic information. This basic distinction of causal and semiotic information is relevant to the question of meaning because meaning is founded in the sign meta-relation. Signs are always signs of. They introduce aboutness in the world by virtue of this fundamental nature, introducing at the same time what we connote by all the synonyms named above, content, semantics, and so forth. Signs are representational in the exact sense of representing one thing in another. This is what signs do. It's not what causal information does. Signs are not the same as signals in the way this word is loosely but pervasively used by biologists. A signal is non-semiotic and usually comes down to a threshold mechanism in the operation of causal information. It doesn't represent one thing in another. A meta-relation is required for semiosis most basically because a sign is something different than a cause of its object. It is instead an analysis of its object. It divides up the object, picking out certain aspects of it to use in representing it. It's complexly constrained in the picking both by its own nature and by the nature of the object. Signs and their objects never mesh completely. That would collapse the sign into the object, collapsing at the same time aboutness and content. There's no representing in identity. But what is it that enters into this complex relation between aspects of one entity and aspects of another? What is the added something that creates the meta-relation? This is a third entity, one capable of forming percepts of certain sorts. It doesn't exercise sovereign control over these percepts. Instead, it's called into the aspectual constraints of what will become sign and object, called by their nature as affordances in the world. Its percepts are always formed not only by its own capacities, but also by the entities that come to form sign and object. Semiosis is in this way a process of reciprocal constraint, reciprocal affordance, if you like, among a percept and things or phenomena external to it. It's a niche constructive process. Those of you familiar with the philosophy of Charles Sanders Peirce will recognize in this approach to semiotics two basic Peircean tenets. The first is the partial aspectual nature of all the relations in the sign-making process. From his scrutiny of this partiality, viewed in its many combinatorial possibilities, sprang the many typologies of signs that Peirce offered. The second tenet is the calling of a perceiver into the aspectual parsing of the world, an idea for which Peirce coined the term interpretant. This is a fundamental term in his philosophy, but one that has been much misunderstood. It involves no interpretation, as we tend to use that word, but relies instead on the perceptual capacity to divide up the world, to parse it, as I've said, registering some of its parts, clumping some things together with others, and relating itself to those relations. How do some life forms come to be able to form interpretants? Memory is crucial, but not merely the simplest forms of storage and retrieval. Instead, something like an episodic memory is necessary, a mechanism that can retrieve memories as parts of whole situations, retrieve them more readily because of those situations, and enrich and relive the situations in the retrieval. Some animals besides humans are capable of such episodic memory. Most animals and all other organisms are not. Learning is also central, but learning of an advanced sort dependent on episodic memory. Simple associative learning is a purely relational thing and occurs in many, many creatures without any semiosis, as a function of simple neural nets involving excitation and inhibition connected with innate reward or punishment signals. A well-studied example is the sucrose sensitivity of honeybees, on the basis of which they learn complex associations concerning food sources and much else. This is non-semiotic learning. On the other hand, the learning of a songbird, for example, to rank the varied challenges from the songs of its neighbors, or to sing by itself a pseudo-duet, suggesting to other birds that its mate is present when it's not, these are semiotic through and through. They depend in each case on the retrieval and processing of whole episodes from a bird's life. And from these, they parse aspects that conduce to interpretant formation. Memory, learning, and finally, attention. Attention is a more basic capacity than memory or learning. And it too comes in more and less complex forms. It's related, even in its simplest forms, to the salience I mentioned before, the way an organism can single out a particular stimulus from all those coming to it. The sensitivity of honeybees to sucrose again provides a good example. For semiosis, a more complex attentional capacity is required, one that can sustain a recursive analysis of incoming stimuli, taking them apart into their aspects and focusing on them at both comprehensive and partial levels. In this analytic or parsing function, attention enables the construction of episodes in memory and learning, building memories as aspects of broader situations. Studies of such complex attention suggest that it's the product of mediations of information in which salience filters proximate to sensory input interface with further processing and modulation downstream. Interface, that is, with top-down systems higher up in the midbrain and forebrain. Such neural mediations are, I think, fundamental to the emergence of semiotic information, and I'll touch on them again later. Attention, memory, learning, these foundations for semiosis are not, as I said before, discrete properties of certain animals, but instead processes in their engagements with their niches. Recognizing the flexible complexity of these processes is what has recently radicalized the ideas of niche construction. Let's step back for a moment. In general, niche construction refers to the reciprocal or feedback process by which organisms build their selective environments as those environments build them. Niche construction has tended to be thought of as a dynamic operating across long timescales, with environments and populations of organisms slowly shifting in tandem with one another. The ancestors of beavers, for example, gradually adapting to aquatic life, even as their behaviors created more and more aquatic environments. Alterations in the niche brought about by organisms bring about shifting selective dynamics, which eventually alter the genomes of those organisms. Now, what I'm terming radical niche construction involves recognizing the pervasive plasticity of niche constructive feedback across all biotic scales of time and space. We now see that niche construction is not only a long-term process, but also the plastic responsiveness of all organisms to their changing environments, operating at a much quicker pace. Natural selection has resulted not in fixed capacities with discrete advantageous functions, so much as in the flexibility by which the nodal systems that make up organisms open out to myriad aspects of their environments. And this is most important, this plasticity reaches from the niche all the way to genes, which turn out to be nothing like blueprints for life forms, but instead are open information systems in constant flux with the situations around them. An example can come once again from honeybees. Dozens of pheromones are crucial in the intricate regulation of the honeybee colony or superorganism. They're produced, of course, by individual bees according to complex bioinformational pathways involving genes, RNA transcription of various sorts, proteins that genes construct, and more. But the pathways run in both directions. The genetic instructions are regulated by the proteins they produce, which in turn are regulated by the external circulation of the pheromones they help to produce. Brief exposure to a single chemical in the cocktail that functions as an alarm pheromone alters factors inducing RNA transcription in the olfactory centers of the bee brain, allowing genes to be expressed that activate fight or flight and aggression pathways in higher brain regions, which then modulate bee behavior. An hour after such exposure, the resulting information cascades have up and down regulated the expression of hundreds of genes, laying a foundation for heightened responsiveness to subsequent alarms. The whole network, extending from niche to genes, has been altered in the blink of an eye, or the whiff of a scent. Similar cascades regulate, it is now clear, the genomic expression of all life forms. With such radical niche construction in mind, let's return to semiosis and ask where we might seek neural substrates for the interpretant and the meta-relation. I think we need to seek them not simply in the redoubling of parallel processing in large brains, and not even only in the serial extension in such brains of processing along a sequence of distinct modules, though both of these are probably play a role. An additional element of neural architecture is built on top of these, bringing massive parallelism and serialism to bear in modulating the processing of other connected centers. Instead of peripheral sensory neurons connecting directly to the highest processing centers, large vertebrate brains have processors connected to processors, all reconnected in loops of feedback systems. The song system in a songbird's brain provides an example. This is often divided into three systems: two pathways controlling song learning, memorization and production, and a third pathway for auditory production. Auditory processing. The slide I'm showing you divides these into two sagittal sections of the brain, with the vocal pathways on the left and the auditory pathway on the right. It's easy to see that the systems are networked in immensely complex ways, involving many nuclei, each with large numbers of neurons. The auditory nuclei are linked to the song control ones, linking left and right-hand diagrams, in other words, and bringing audition directly to bear on memory and learning. Especially important for the semiotic complexity of birdsong, I think, is the mediating role of the thalamus in these networks, situated between mid and hindbrain and higher cognitive functions. Notice that it doesn't merely relay process sensory input to higher brain regions, but is a part of loops from those regions involved in both song production and audition. In mammals, the thalamus is a midbrain processing station mediating top-down and bottom-up networks. It's implicated in attentional focus, voluntary motor behavior, memory, and learning, and so it seems to be in songbirds. There's nothing nearly as elaborated as these networks in the brain of a honeybee. Here, by contrast, is a diagram of the network for sucrose sensitivity and responses that I mentioned before. The relevant part of this diagram is the red arrow, tracing a single long neuron that projects from the proboscis through the antennal lobes to higher brain regions, the so-called mushroom bodies, and to the green motor control center. The complexity built into this tiny brain is truly awesome, but is far outstripped by the bird brain's multiplicity of distinct modules, their serial and parallel connections, and their intricate feedback mediation through distinct levels of processing. It's not merely the complexity of neural architecture, however, that brings about the cognitive functions of interpretant, sign, and meaning. To achieve this, we need to set this network in motion as a process rather than an architecture. And the motion comes about through the radical niche-constructed plasticity in the face of changing affordances I spoke about before. The informational complexities I exemplified with the alarm pheromones of bees, altering gene expression on a moment-to-moment basis, are at work in all animal brains and extensively studied in songbirds. They seem to foster a varied and complex processing of the world proportionate to the varied complexity of the brain architectures they enliven. It's from this, I think, that a new fold is introduced into the experience of certain animals. From this, that they gain the parsing power that makes their percepts analytic, and at the same time opens those percepts to the formative rebound from the things they analyze, the calling into the interpretant. From this fold, this recursive percept arises the cognizance of a relation in perception to a relation of things in the world, the essence of the sign and meaning. Let's return finally to the challenges facing the project of an AI that could construct and understand meaning. What should be apparent from my quick overview of the foundations of animal meanings first is the wild over-optimism of Rodney Brooks's assertion around 1990 that his robots were approximating even insect levels of intelligence. The complexities of bee behavior depend on modest attention, memory, and learning, all rendered processual and reactive to the changing world by systems of gene regulation linked through cellular pathways out to external stimuli. In insects, these are all causal informational complexities. They do not manifest the meta-relation or create semiotic information. So the first step in making a thinking machine would be to come somewhere close to the complexities of radical niche construction evident even in non-semiotic creatures, complexities that are still not fully appreciated 30 years after Rodney Brooks's embodied, and situated AI. This would give robots an ability to react to their environments in ways that resemble the plastic genomic shifts of living organisms. This is a very high bar for machinic intelligence. Even surpassing it, however, wouldn't endow a machine with the ability to create aboutness and meaning. For this further leap, we'd need to marshal systems of the sort exemplified by our songbird, and then harness them to the plastic capacity animals show to remake the systems in moment-to-moment negotiation with their niches. The machine would need complex attentional capacities meshing high and low-level processing. It would need episodic memory, recalling whole situations in its past and shaping its learning on the basis of them. And all these systems would need to submit themselves to a ceaseless alteration on every scale, starting from the smallest generative ones, the genes in the machine, so to speak, which could not be conceived as fixed entities. Now, I'm not an expert in AI or indeed any kind of computation. My only programming experience was a course in BASIC I took 50 years ago at Dartmouth College, taught as I remember it by John Kemeny himself. Or perhaps he only visited the class, leaving its day-to-day operation to his graduate students. But I pose the question to those who do know about AI. Are we coming close to all this? Can all the evolutionary algorithms, deep learning, and meta-heuristics of today's AI begin to glimpse the lifeways of animals that bring about meaning in the world? Can they approach even a lesser standard, the autopoietic intelligence of a honeybee? Thank you very much.
Our visual system is amazing. We can make sense of the world around us just by analyzing the light that bounces off objects and reaches our eyes. To illustrate this, let me show you a movie. This will help you to appreciate how amazing we are. This following video is showing a very low quality movie recorded in my office. This movie is so blurry that many of the objects are defined by only one single pixel. But our visual system is so amazing that can recognize a person talking on the phone, listening to some music, and working on a computer. And it is amazing that we are able to recognize so much from so little visual information. Of course, most of what we saw in this video was not really there. Most of what we saw in the video was only inside our heads. And to illustrate this, let's watch the original high-resolution movie. As you can see now, many of the objects that you recognized before were not really what you thought. So why is it so difficult to build a machine that sees the same ways that humans do? Well, the input to a vision system, an image, is just a collection of numbers like the ones shown here. Each number represents a pixel intensity at that location. The numbers do not directly tell us which objects are in the picture or even which numbers are part of an object or part of the background. This is what a machine will see, and what we see here, if we look at the same numbers but displayed on the form of a picture, then we see just a blurry picture of a person walking. Computer vision systems need to be designed and trained to see the person behind the numbers. And that's where the challenge resides. Training a system capable of recognizing objects in images generally requires annotating manually thousands of images where we will have to indicate what are the objects in each picture, their boundaries, and what the name is. And the system will then learn to associate the image with the annotations that we provided. And this process is very labor intensive, and it requires many hundreds of hours of manual labor. A different strategy that is currently used in research is to train systems using computer graphics. So for instance, the community is developing virtual worlds where you can interact with agents, you can see the world, recognize objects, and train AI systems in these virtual environments with the hope that later they will learn something that can be transferred to the real world. But all these different ways of training artificial vision systems are very different to how babies actually learn to perceive the world. And so let's watch a baby actually learning to recognize things in the world. A baby learns most of the time by itself. There is no supervision from the parents. There are no labels. And it learns by interacting with the world, by looking at things, by hearing the noise that they make, by touching them, and finally tasting them. And a baby will learn to discover objects, to understand materials, to perceive a space, to manipulate objects via interaction. And it will do this through a very rich set of sensory modalities: vision, audition, smell, taste, and touch. And it does this in a complete unsupervised way. It doesn't require the type of exhaustive annotations that current AI systems need to learn about the world. So in this presentation, I will describe some of the work in our groups trying to build systems that leverage different sensory modalities to learn to perceive the world without supervision. So we started our work in 2016 with a very simple setting. Andrew Owens, a PhD student at MIT, recorded video and sounds while he was hitting surfaces with a drumstick. For over a month, he walked around with the camera and a drumstick and recorded many thousands of hits. So let me show some of his greatest hits. He hit it and scratched surfaces all over MIT for a long time, and he recorded tens of thousands of these events. So with this data set, we asked the following question. Given the video input, without the audio, just the pixels, is it possible to predict what the audio should be? So we want to build a system that will take as input just the pixels, just the frames of the video, and it should produce the sounds that should go with it. A system trained to solve this audio prediction task should be able to solve the following problems. It will have to recognize the drumstick, and it will have to detect when it is in contact with the surface and whether it was a hit or a scratch. It will have to recognize the type of contact, the strength and the duration of the contact. It will have to recognize the material where the contact happened. And finally, it will have to associate all of this with the sounds that you hear. So we did that task. We trained a system to solve this problem. And what I will show you now is the result of running that system on a silent video. So what you will see, the input video shows the drumstick hitting and scratching a rock, but the system will only see the images, not the audio. What you will hear as I play this video is the audio predicted by the system. It's not the real audio that came with the video. So let's play this. And as you can hear, the sound was correctly temporally aligned with the moments in which the drumstick was in contact with the surface. And the sounds corresponded to hits and scratches. It was not perfect, but the system shows that it has learned something about the visual and auditory worlds without any human supervision. So what has the system learned? The network has learned to represent surface properties from visual information only that allows to discriminate among different types of materials. A simple classifier trained on the self-learned features can discriminate among different types of materials. It will confuse materials that produce similar sounds when hit with a drumstick, such as glass and metal, which sound very similar when you hit them with a drumstick. But learning with sounds and images can be done even without having to hit everything with a drumstick. In fact, when we are in a particular place, anywhere, we will always hear some sounds. The ambient sound that surrounds us is a mixture of all of the sounds that are made by the objects that are present in the scene. So it's like thinking that the world comes with its own soundtrack. And many objects in the world produce sounds by themselves of different kinds. And the main cue that we could use here is the temporal coincidence of hearing a particular sound and seeing a particular visual pattern in the world. And then we can create an association between that sound that we hear and that particular visual pattern and the fact that they seem to co-occur time over time. So we can now play the same cross-modal prediction game that we did for the drumstick. We will try to predict what type of sounds are associated with a particular image. And in this case, the image will not contain the drumstick. It's just whatever objects are there that are producing sounds. And a model trained to solve this task, if it is successful, it will have to solve the following problem. It will have to be able to recognize and categorize the objects that are in the scene that are naturally associated with ambient sounds. And it will have to associate them with the appropriate type of sound that it goes with that object. So what has a network like that learned? We can now, once we train such a system, we can look inside what the system has learned. Just in a similar way of how a neuroscientist will look inside the brain, look at the activations of the neurons to try to understand how the brain works. We can perform the same thing on a neural network that has been trained to solve this task. And what we discover is that the network, inside the network, some of the neurons became specialized in detecting baby faces. Other units became specialized in detecting crowds of people. And this is because babies produce a particular type of sound. And whenever a baby face was present, that was the sound that was associated with it. Crowds also made a different type of noise. And there are other units that are selective to waterfalls, the ocean, and all of those units have learned to detect objects in images that are associated with distinctive types of sounds. There are units that will detect also the sky because it comes with the sound of the wind, and also cars. The important point here is that these units have learned to detect these objects without requiring any type of human supervision, just by learning from the temporal association between sounds and visual patterns. In particular, for instance, the car selective unit is capable of detecting cars, but it has never been trained with a human having to annotate cars in images. Sensing images and audio has a long history, but there are other types of sensory modalities. And in particular, one very important sensory modality for humans for which do not exist a mature technology is touch. Tactile information is essential for humans, with a significant part of our brains devoted to process tactile information recorded by our skin. And there are a number of devices, commercial devices and research devices that have developed to measure tactile information. But it's still a very small number. Those devices are nowhere near the quality and maturity that cameras and microphones have reached. We are developing tactile sensors that will also record tactile information when a human interacts with an object. In this particular case, it's a glove that a human can wear that records pressure signals. And as you are manipulating an object, it will record which parts of the hand are in contact with the object and what is the strength of that contact. This way, we can develop approaches that can use this measured tactile information to perform object recognition, to measure object properties, and so on. And it's not just about the hand. The same technologies can be extended to cover large body surfaces and eventually become a tactile sensing skin for robots. So as I mentioned, all these different sensory modalities is what provides now a way of learning from the temporal coincidences between different events across different senses. And this will provide a method to learn about objects without requiring human annotations. Besides interacting with the world, there are many other ways in which we learn to recognize things. So let's think of a following game. Let's say that I give you lots of images and you have to play the following game. Given a collection of images, I want you to draw a new image that looks similar to the images that I gave you. It doesn't need to be one of the images that I gave you, but it has to look as belonging to the same family. Playing this game requires understanding what the content of these pictures is, understanding the 3D geometry, and then drawing something that looks similar to it. And current systems current neural networks have been tackling this problem for some time, and there has been a lot of progress in recent years. There is a family of systems called generative adversarial networks that try to solve that problem. Given a bunch of images, they want to be able to draw new images that look real. This approach was proposed in 2014, and these are the type of images that were produced by an artificial system trying to reproduce images that seem and look real. And as you can see, they were very low quality, very similar to the blurry movie that I showed you at the beginning. 2 years later, new advances were capable of drawing images like the ones that I'm showing here, which are trying to look as bedrooms. They are not very good, but you can see that there has been a big step forward and that there are some geometric figures that look like they could come from the real world, although no one will get fooled in thinking that these are real images. But just 2 years later, the advances were so big that systems were capable of producing images that look like this, and therefore quite real. And since then, there has been a lot of progress. So the system, what it does, it starts with a collection of images like these ones that contains buildings, churches, and castles. And from this collection, it will train to be able to generate new images that should look real. The way in which this neural network works is it will take as input just a random number, a random collection of numbers, like flipping a coin 500 times and recording just the outcomes. This vector will get enter into a neural network that will recombine this set of numbers, make it bigger, recombine again, enhance it, recombine it, until at the end, you end up with a picture. And this picture is a randomly generated image. If you throw the coin multiple times, different times, you will get different set of images. So these are examples of images that are not real images. They are generated by this neural network, and they look amazing. They even have watermarks because they were part of the training set. But the question is, to render images that look so real, what is that this neural network has really learned? Does it know about objects? How it is capable of putting the pixels in the right places so that they look as sky, trees, and buildings? Is there a representation that is explicit and it contains some of these objects? So that's a question that we ask ourselves, and we started looking inside the neural network to see if it was discovering some of these objects. And indeed, what we found is that there are units inside the network that have learned to draw specific objects. So in particular, for instance, in a particular layer number 4, neuron 119 became a tree-specific unit. Whenever that unit was activated, a tree will appear on the picture. Another unit number 43, whenever that neuron was activated, a dome will appear on the picture, like in these images. There was another neuron 84, that was specialized in drawing windows. And whenever that neuron was activated, a window will appear on the picture. And unit 315 was specialized in drawing chairs. And whenever that unit will be activated, a chair will appear on the picture. So what is really interesting is that these units that are explaining some objects appearing or disappearing on the picture, now we can intervene on this neural network and control what we want to have on the picture. So for instance, this image, which is randomly generated by the neural network, if we take the units that we know are responsible of drawing windows and we turn them off, we tell them not to get activated anymore in this picture, and then we render the resulting image, what we get is something like this picture which is very similar to the other one, but it doesn't have windows. And instead, it has replaced the window with a wall, and there seems to be a painting on the wall. And the only thing that we have done is to manually turn off the units that we believe are responsible of drawing windows. Here is another example of a different image that is drawing a conference room. If we turn off the window units, the windows in the background disappear and they get replaced by diplomas, which seems to be an appropriate decoration for a conference room. All these images are completely randomly generated by this neural network. We can also ask the neural network to do things that are impossible. For instance, this picture is very gray, and if we want to have more color, we can turn on neurons that we have found are responsible of drawing grass. But we are going to activate those neurons everywhere on the picture, and then the resulting is something like this, where it seems as if humanity is gone after 1,000 years. And you can see that grass is growing everywhere, is growing on top of the building. And these are things that this neural network never saw during training. It's putting objects in places where we are asking it to do it. But this is something that the network never saw during training. So the network seems to have learned to represent objects explicitly, despite that, again, there was no human annotations. No human had to indicate what a window is, what a car is, what grass is. The network has learned to associate parts of the network with specific objects. So this is part of this enterprise of trying to learn without supervision, discovering what are the objects in the world through different modalities and imagination, so that you do not require a teacher or human annotators in order to train an artificial system.
My name is Markus Bühler. I'm the McAfee Professor of Engineering at MIT, and today I'll be presenting some of our recent research on the molecular mechanics of unfolding Johann Sebastian Bach. We all know Johann Sebastian Bach as an influential composer that is still affecting musical creation to this date. But have you thought of Johann Sebastian Bach as an inventor of new proteins, as a creator of new biochemistry? We have discovered that Bach might have inadvertently created, designed, and realized new forms of life, new forms of proteins, new biomaterials that evolution has not yet discovered, but that have been created through the creation of musical art. This provides evidence for hidden information created across timescales, created across cultural information levels, to inform science today and perhaps the future of understanding life on this planet and elsewhere. When you think about the formation of the fundamental building blocks of life, you begin to think about proteins. Proteins really are the elementary ancient evolutionary memories of a very distant past during which evolution yielded the kind of life we have on Earth today and that emerged over millions and billions of years. Proteins spontaneously come together or fold at very short timescales, beginning with timescales of 10 to the minus 15 seconds, which are the scales at which molecular connections vibrate. On the other hand, on the other side of the spectrum, we have human-created assemblies of structures, such as music. Music is an assembly of pitches, notes, different timbres or instruments, in complex arrangement, in complex hierarchical arrangements that span multiple time and length scales. Through the assembly of notes, we can induce emotional responses in our brain. Through music, we can convey messages, either direct messages, obvious messages, or subliminal messages that are underlying currents in the musical creation. With the new tools of artificial intelligence, we can now make new connections and discover hidden physical realizations of information in art to advance science, humanity, and culture. There are structural forms in art that we believe can only now be discovered using modern technologies. Our research deals with multi-scale materials. We look at matter from a multiple scale perspective, from the atomic scale, where we have dimensions of 10 to the minus 10 and smaller meters, which are the scales of atoms and orbitals and chemical connections, And we go all the way up to the macro level, where we have scales at the human level of a couple of meters, or at the scale of the planet, many, many thousands of meters and kilometers, or to the scale of the universe, where we have even much larger length scales. These materials can be looked at through this microscope of different magnifications. And at each level of magnification, we're going to see different structural features. These structural features range from the atomic scale through the micro, mesoscale, all the way to the macro scale. And materials derive their functions, their unique properties, such as color, mechanical properties, optical properties, magnetic properties, diffusive properties, and any other kind of technologically important or culturally important properties through these features across these different scales. These are patterns that humans in part have created, or nature creates all the time during evolutionary processes. One of the fundamental processes we're interested in is the breaking of materials. Materials break in characteristic timescales of 10 to the minus 12 seconds. There's a universality of vibrations and waves, which occurs at every observational level that we have currently available. We have molecules which can be understood based on a wave-particle duality. Quantum scale objects behave either as a wave or a particle simultaneously. We have sound waves, of course. That's how we communicate. That's how animals communicate. And we have used sounds in the form of music as a cultural expression, as a way of affecting memory and passing on information through generations of cultures. And of course, we have the insect world. Spiders, bees, and wasps all communicate through vibrations. With tools we have now available of artificial intelligence, molecular dynamics modeling, we can map these different timescales on a single observational level and actually make them understandable through new processes in the use of the human brain. In the beginning of the universe, we had a fairly even distribution of matter. Small fluctuations of matter ultimately led to the creation of planets, galaxies, and all the things we have today, including atoms, of course. Localization of information, localization of energy or matter, which are equivalent, leads to the creation of matter as we know it today. In a very similar way, when we look at spiders as an animal that creates an extra-organismal structure, the spider web, it also does this using a very similar mechanism. A spider might eat a fly. The spider's world consists of eating things, processing the things it has eaten in the body of the spider, and then reassembling these building blocks of what it has eaten into a structure. From a distribution, a random distribution of protein building blocks that the spider has digested from eating the fly, it can create beautiful web structures as shown here. And at the foundation of these web structures, of course, are these protein molecules. And these protein molecules are made by themselves from what we call amino acids, or ultimately encoded by DNA. And these can be viewed as Lego building blocks. So when the spider eats a fly, it essentially ingests a very large number of Lego building blocks, and it has a random collection of these in its body. And the spider has found an ingenious way to then reassemble these building blocks, these Lego building blocks, into a structure, to organize them in space and in time to create a certain material function, which in this case is the web's function. One of the functions is to catch more prey for the spider to survive. Here's the result of an experimental analysis where we observe the spider building a web. This is work in collaboration with our CAST collaborator, Tomas Saracino, with whom we watched the spider build a web over a period of days. And you can see how the spider is truly an autonomous 3D printer, depositing materials in space, choosing the location, and thereby creating a hierarchical structure that is both aesthetically beautiful, but also, more importantly, for the spider, functional. And in a similar way, when life emerged billions of years ago, we're looking now at the evolutionary time scales, we had random processes, random fluctuations that created structure, the basic structure of the formation of RNA or other molecules, which ultimately created life as we know it today, built from DNA, proteins, and other kinds of biomolecules. And this led to the creation of all sorts of different organisms as we know today. And again, these organisms have evolved because ultimately, at the basic level, molecules assembled in particular structure from a random distribution of molecules, a barren landscape that this planet was billions of years ago, where then suddenly molecules began to arrange, became living objects, living subjects, which ultimately led to the creation of the human race as we know it today. Music, in a very similar way, is an election of localization of energy. This election is created, for example, through the creative mind or other processes, or animals which use sound or music to communicate. White noise is the equal distribution of tonal energy at all frequencies, creating a chaotic, a random structure. And we represent this in our ear and our brains as noise. This is in stark contrast to what we identify as harmony, where instead of having a random distribution of information at all frequencies, we now have a distribution at very particular frequencies. By playing tones at very particular frequencies or a selection of frequencies, we can create musical information and encode information and create an impression in our brain that causes an emotional response. Here's an example where I begin playing white noise and then use this white noise as a basis to create sound that we realize in our brain as music. I use the white noise and then filter out only the harmonics. These harmonics are the building blocks for very simple forms of music. And you can hear as the sound progresses, we go from white noise, filtering out information and only localizing energy at these harmonic frequencies into a single tone. We can then play music on the form of octaves by using equal temperament tuning and even create chord progressions or other more complex sounds. And as we turn the filter off, we return back to the white noise. So in this case, we have created a random structure and we have localized information, we have localized energy and created what we call music, or one of the forms of music that we know as today. Another way to create music is to start from silence and only create tones or sound that reflects certain types of frequencies or vibrations. This is a localization of frequencies that we typically use in the conventional way of using analog instruments, like a vibrating string. These vibrating strings vibrate at very particular frequencies. We can tune them by changing the tension or the length of these strings, and we can then create music by having different types of tuning systems and plucking the strings differently or bowing them differently. And of course, an example for highlighting the emergence of sound or music from silence is the famous piece 433. The strings don't have to necessarily be conventionally tuned. They don't have to look like in a guitar or in a piano or perhaps in a violin or viola or cello. These strings actually can also come from nature. And in some recent work, we have used spider webs and used the individual strings in spider webs as musical form, as musical creation process. And you could imagine that these strings in the spider web are not tuned according to any of the tuning system we're usually familiar with. In fact, they're tuned by the spider to follow certain rules and laws that the spider uses to communicate. But we can make them audible. And what you hear in the background is an example of how these types of vibrations sound like. It's a strange world, but you can begin to live in the world of the spider and engage in the process of feeling a little bit like a spider. Music changes the state of mind, and it has affected our physicality. So when you listen to music, when you hear sounds, you have a response in your brain. It's a physical, real response. Things are changing in your brain. Things are changing. Material changes are happening as you listen to music. Before and after is different. Our question now is, can these material changes yield actual new material form? Instead of changing our brain, state of the molecules in our brain, the chemicals in our brain, the cells, the neurons, can we utilize music as a way of creating new material form or perhaps new life forms? For example, can we create new biomaterials? Can we create new types of cells? Can we create new proteins? This has been the topic of my research in the last couple of years. And we asked the question, how can we bring resonances and motions from the distant past, perhaps from composers that have composed music, created music hundreds or maybe even thousands of years ago to the future, and how can we analyze them, understand them, and actually use them to create new kinds of material expressions and extract information that's not obvious, subliminal information that's hidden in the musical structure. We can use this approach to better understand materials. We can use it to define new musical forms as a new way of composing music, of creating music. And we can also learn about ourselves, because this music is created by humans. And by studying what humans create and what hidden messages we might be sending, we can communicate across generations. So again, you have a multitude of length scales and time scales involved here in this process. Proteins are the hardware of life. An introduction to proteins essentially begins with the level of DNA. DNA is essentially the coding structure. It's elementary to understand how life is built. It's the building plan for that. DNA encodes the way proteins are actually physically constructed, how the amino acids in proteins are put together. Ultimately, the protein folds at these micromillisecond timescales to form ultimately living subjects. And these living subjects have functions. They form perhaps cells in a body. They form organisms, organs. They can form tissues, our skin. You can think about your eyes. All of these things are really truly made from proteins. So proteins are the workhorse, the actual materialization of life. And in the evolution, this plays a very important role. Proteins are evolving, of course, through genetic mutations, through natural selection. And these processes lead to different life forms. So all these different life forms have different kinds of proteins. Some proteins have survived evolutionary processes and are found across different species. Other types of protein molecules are only found in one species, or maybe in one particular niche ecologically. Now, how do proteins look like? When you open the textbook and you look into molecular structures, you're going to find a picture like this. And you're going to find a chemical structure, usually out of water molecules, maybe some metal ions, and really reflecting a complex folded geometry, and thereby deriving chemical, biochemical, and evolutionary functions. But if you think about proteins and molecules in general, that's really not how they look like. If you take a real microscope and you watch the molecular motions, which now we can actually image experimentally, you can see that these atoms and molecules are actually continuously moving and vibrating. As these molecules continuously vibrate and move, they follow equations of motions which ultimately are governed by quantum chemistry. These quantum chemical laws can be implemented in an algorithm called molecular dynamics, and we can actually watch, simulate, and observe the motions of these molecules. These vibrations happen on the scale of 10 to the minus 15 seconds and higher. So these are very, very short time scales compared to the scale of observation of the human mind, for instance. And if you look at a simulation of a protein, this is an alpha helical protein, you can see that these vibrations, these jittering motions, are quite interesting, quite beautiful, very elegant, and you can see they have certain patterns with them. And these patterns are associated with certain structural features. We can distinguish different kinds of proteins by watching different kinds of features or patterns emerge at these very, very short time scales. Now, these vibrational signatures are the elementary description of life. Proteins are made from 20 naturally occurring amino acids. And each of these 20 amino acids we've discovered has a unique vibrational signature. They vibrate in different forms, in different ways. And these vibrational signatures are sort of barcodes for the elementary building blocks of life. And this represents a localization of energy. Just as we talked about it during the creation of the universe a very long time ago, during the emergence of life, during the creation of music through the human creative mind, in all of these processes, there's a common thread, a common theme that we localize structure. We put things in a certain place, remove things at the other place, or we don't even put that at the beginning. And thereby we create a distribution, a patterning. And the way life emerges is creating patterns across these 20 amino acids. Now, these 20 amino acids can be made audible. This information is stored at very high frequencies, and naturally we cannot hear them. Our auditory system can only hear up to about 20 kilohertz. We can solve this problem by using the concept of transposition or transpositional equivalence. As this simple example shows, I'm beginning to play a melody in the beginning at very high frequencies, and I'm slowly moving down. I'm transposing this piece of music into lower and lower frequencies until it becomes audible. So as you listen, pay close attention and see at what point you actually recognize what melody I'm playing. Once you have recognized the melody, I'm playing it again. I'm transposing it again to lower frequencies. And to your ear, to your brain, you can clearly still identify that this is the same melody. It's Four Lieds by Beethoven. However, I'm playing it at yet lower frequencies. And this is what we call transpositional equivalence. We can play melodies at different frequency levels by keeping the ratios, the multipliers of frequencies, the same. And thereby we retain musical information, structural information that is elementary to understanding through the auditory system in our brain what kind of sound has been created and what that means for our processing in the brain. In a similar way, we can use this to translate proteins into audible spectrum. And what you hear in the background now are all these 20 amino acid building blocks played in a succession. And you can hear that each of these 20 amino acid building blocks has a unique tone associated with it. It's kind of like having its own key on a keyboard or a piano. However, this is not a typical piano because the tuning system, of course, now is a different one. It's a distinct tuning system that we call the amino acid scale. It follows the quantum mechanical laws by which these 20 naturally occurring amino acids actually vibrate. We can thereby map proteins to sound. What you hear in this example here is the mapping of a protein called lysozyme. It's an enzyme that's been studied for many decades. This protein has been translated now through this process into a musical piece. In this process, we also recognize the folding of molecules by playing melodies against each other, creating a sort of counterpoint emergence, a counterpoint function that is very similar, for example, to Brian Eno's work using tapes. So these tapes, you can imagine, are the amino acid or DNA sequences that are folded on top of one another, thereby forming more complex musical form than just the sequence. And we also incorporate, of course, higher level structural information, such as the vibrations at higher length scales and time scales, as well as secondary structures. These are the distinctions between an alpha helix or a beta sheet. Each of these have unique auditory signatures which can be made audible. So we're creating a symphony of auditory information that encodes the three-dimensional structure of a protein from the very basic level of amino acid sequences all the way to the structural, the folding scale. And this can be applied not only to the amino acid scale, we can actually approximate the amino acid scale using conventional tuning systems such as equal temperament tuning and convert these types of protein sounds, these kinds of protein music, into a form of Western classical music and play them using an orchestra. And we've done this recently in a piece where we have sonified the antibody to the COVID-19 pathogen that forms when you take a vaccine or that forms when you're naturally ill from this virus. Your body produces these antibodies which interact with the virus. And these vibrational interactions, this system of vibrating atoms and molecules that interact the virus with the antibody trying to defeat it, is what you hear in the following example, which is set in a classical score, reflecting the information in this particular protein system. This piece has been played by the Lindenbaum Orchestra in Korea, who have realized this in an orchestral setting. Why are we interested in translating proteins to music beyond the aesthetic approach, beyond maybe as an interesting way of creating new musical form, creating new musical competition to inspire us to create interesting musical structures? We can also use this to design new proteins. We can create training sets for artificial intelligence algorithms from this process. Imagine having a large database of proteins, thousands, hundreds of thousands of proteins, which we can then translate into music. We can let an AI algorithm listen to all of these proteins and let the AI compose new music. This new music can then be translated back into a protein structure. This approach has been very successful, and we have used this to actually make new proteins in the lab that we've designed through music. I should mention that instead of having an AI create music, of course, we can also have a human composer create music and write music in the same form or style as these proteins produce. So we can learn these processes. We can learn the patterns, the rhythms, the folding, the interactions, the counterpoint structures, and utilize these to thereby create new proteins. But using AIs, of course, is a perhaps simpler way because it's not influenced, not biased in any way by the kinds of musical impressions that we all have by listening to all the other music we've ever heard in our life. This is the way by which we translate music into a new kind of protein, a new kind of material. We begin this process by creating a musical score. These musical scores include all the information about the notes, the volume, the rhythms, and the counterpoint information as well. And then we translate these sequences into a sequence of DNA building blocks. This DNA sequence then forms the basic instruction set for cells. In this case, we've used E. coli cells to then express this protein. Then the cells make the protein, and we can basically slice the cell open and extract the protein and purify it and put it in a test tube. And what you see on the right-hand side is how such a test tube looks like, which includes this protein that was designed purely based on music. Now, this protein is what we call de novo protein. It doesn't exist in nature. So through this musical approach, we have discovered, instead of using evolution as a way over a very long time scale, we've used in a very elegant way musical creation as a way of designing and realizing in material form a new protein. So now we're going to do an experiment. Let's listen to the beginning of the aria in Bach's Goldberg variation. And the question is, what kind of information is stored in this very beautiful but very simple succession of notes, of rhythms? And by analyzing this music that Bach wrote a couple of hundred years ago, we can assign each of the musical notes in the aria, the beginning of the aria, into a succession of DNA codes by assigning unique associations with these vibrations of amino acids. This DNA code can then be formed as a basis into a protein expression, just like we've done before in an experimental way by putting these amino acid sequences or DNA sequences into a cell. In this case, we've put this into a deep learning and machine learning algorithm. These machine learning algorithms have the capacity to predict how these amino acid building blocks fold or assemble at these micro to millisecond time scales and form a material. And these algorithms can also predict, of course, whether a material will form or not. And that's a big question because some of the successions of notes or rhythms will not ever form a protein. There are certain rules by which these musical creations in this protein amino acid scale must follow. And the experiment here is manifold. One of them is, did Bach actually inadvertently create a protein that can actually be folded and is stable or not? And the algorithm has predicted a very surprising result. So we used this deep learning algorithm to fold the protein, and we discovered that, in fact, the first 146 notes of the aria indeed lead to an actual protein that is stable. And you see on the right-hand side how it looks like. So we went from the musical form that you could hear into a ladder of DNA sequences, protein amino acid sequences, to ultimately a protein structure that we have folded, and you can now see and watch. Now, this protein is sort of the realization of this music. So I have on this slide a comparison of the musical score on the very left, the original musical score, and on the right-hand side you see the protein. You can see again on this example here that the protein isn't static. As I mentioned at the very beginning, a protein like this is continuously moving and vibrating, right? So these atoms are not sitting still. They are like mini strings, mini meaning at the scale of 10 to the minus 10 meters. So very, very tiny strings or connections between the atoms, the chemical bonds. They vibrate and they have a certain signature, a certain vibrational signature. Now, what we've done on this experiment, we've taken this protein that we've created based on Bach's composition and translated this protein again into music. So the question is, can we sonify this protein? Essentially, instead of taking an existing protein that evolution has produced, as I've done before with lysozyme, the enzyme, we ask now the question, can we convert this protein created by Bach, inadvertently, of course, into a new musical structure, and how would that sound like? Here, we're going to listen to that piece now. And what you hear in this example is the protein that was folded based on Bach's Goldberg variation aria, the first 146 notes, including all the information about how the protein folds, the secondary structures of rhythm, the counterpoints. You can see the counterpoint map on the right-hand side, and we can see what kind of interesting new music comes out of that process. So what this experiment has shown is that the music that we have created has new information. And the new information has been added by using the deep learning algorithm to really incorporate truly the ancient memory of evolution. The ancient memory of evolution is essentially the building blocks, the laws by which proteins fold from the sequence. And this folding, this chemical information, this basic life information, the living system information has now been incorporated into this note sequence that Bach created, and thereby created new musical information, new sound information that was not there before, and actually has created a piece that is about 5 minutes long in this case. This new protein is different from any protein that has been created by evolution. In fact, we have checked this protein against any known databases of proteins, and it doesn't exist. So Bach has actually created, through this process of writing down notes, new proteins. We can now make this protein in the laboratory and analyze what kind of function it has. Now, the functions it might have, it could be an enzyme, it could be a structural material, in other words, it could be a strong and tough material, like in hair or fingernails. It could be a virus. It could be a pathogen. It could be a poison. It could be a hormone protein. We don't know what it is. And this is where, of course, much further research is needed to understand the exact function of this protein encoded by Bach. But more broadly, this process has also enabled us to create a new way of composing music. So instead of writing down notes, we can write down notes and then ask a deep learning algorithm to incorporate new information, new secret or hidden information in this process. And through this process, we can incorporate information across very, very long timescales and length scales, essentially looking at the evolutionary timescales during which life and all the proteins have evolved. And this creates a new set of musical ideas that we can utilize perhaps in composition or in the writing of future musical forms. So there are a couple of things we have understood or learned from this experiment. One question you could ask, why isn't the music yet comes out of sonifying the protein that we have created from Bach the same as in the beginning? And the answer is that, of course, the folding process has added new information. So we have utilized a different medium, a different expression of information, the quantum chemistry, the evolutionary timescales, and added this information into the folding and ultimately into the listening experience. And so the music has changed after the expression as a protein from the DNA sequence that Bach originally invented. And this is a passage of information through a very different timescale horizon, of course, that includes the vibrations of the atoms, which ultimately make the sound, the music you hear. But the memory that we have incorporated, of course, comes from a very, very distant past. So AI can help us to create art, or in particular music, as we've talked about here in this presentation, but it really does not replace humans. It can be a tool for humans to become more creative, or to get information and answers from a distant past. We like to use proteins as a way of gaining information, musical ideas about things that have happened in evolutionary timescales. We like to interrogate proteins and molecules to see, to hear what is happening at the scale of vibrations of amino acids or between atoms, and looking at atomic bonds like a guitar string that we can excite and watch and hear and use as a way of creating a new scaling system like the amino acid scale. But in all of this process, even though computers and AI can help us, we still need a significant input, of course, and artistic choices by the human, especially once we want to create artistically interesting or aesthetically pleasing products that we can process and understand in our brains. So the creation of music is really a deeply human experience. It's a way of mapping out memory of deep time, and the individual and collective process by which music is created, performed, improvised, exposed on, analyzed on, derived for future evolutions of musical culture, musical creation, is really an unconscious act to create consciousness of the past. And this concept is artistically very interesting. It's also scientifically very interesting, because using music as a way of looking inside ourselves, to looking at these distant primordial memories of structures that might have happened during the emergence of life billions of years ago here on this planet, perhaps even further, deeper in time, during the emergence of the universe as a whole, where quantum fluctuations have emerged. And I think this is a very interesting direction for the current and future research in my lab to explore whether these relationships can be brought out, whether we can make them audible, whether we can connect them mathematically using concepts like category theory to other principles in organization of information, and thereby solve, of course, ultimately important cultural problem or scientific problem, or to advance the ability of humans to create music and to create this beautiful abstract form of art that allows us to transmit information, emotions, without ever using a single word. So the opportunities ahead are manifold. We can see that by looking at multiple scales in a material, you can watch individual molecules move. You can watch spiders build webs. We can make spider webs into instruments. We can make molecules into instruments. We can hear music everywhere, really. We can hear the sound everywhere. And this tradition, of course, of using sonification, or the tradition of listening to the environment, or understanding how silence or the lack of music, the expectation, like in 433, yields this particular result of paying close attention to nature, close attention to the environment, is powerful. And with the tools of AI, molecular dynamics, multi-scale experimentation, microscopes really, whether computational or real microscopes, we can look inside materials and understand a lot of these principles at a very deep and foundational level. And what you hear now in this last example is music created during the fracturing process by analyzing the stress tensor. I'd like to thank you very much for your interest and for your time. Please be in touch by email if you have questions. Follow me on my SoundCloud page, where you can hear a lot of examples of music created from proteins, music created from fractures, and sounds of spider webs. And of course, you can also follow me on Twitter, where I post information about my latest research on a regular basis. Thank you very much, and I see you hopefully soon in the panel.